Okay, we are good.
All right. So, Catalin, this is your beta testers.
Fantastic.
This is going to be Catalin, and coming at us from Vienna. He took the time out of his day to do this and talk to us about those devices that he made and you all got to play with. So, everybody give him a nice introductory round of applause.
Thank you, guys. I cannot hear you very well, but I could hear Mike. I apologize for my voice. I'm a little bit sick. It's not COVID according to the test, but it's something similar. And I guess you can see my screen, yes?
Yes.
So I'm going to talk about Replifactory, the devices that you've been using. And before that, I will start with a small introduction. So what exactly is experimental evolution? That's the experiments that you've been running. There's this nice definition from Nature that says that it's the use of lab environment to look at evolutionary processes. So unfortunately, we cannot look at the evolution of animals such as mammoths or elephants because they grow too slow, but we can do that with microbes, which grow very fast. And the first experiment of this kind was actually more than 140 years ago. A guy named William Dallinger, who lived around 1880, started this experiment by taking an incubator, where he could control the temperature. And in these three jars that were inside the incubator, he was growing 3 unicellular organisms that had a flagellum and looked roughly like this picture here. Can you see my cursor?
Yes.
Yes, you can. Okay. So if I do this, you'll see that. So he grew these unicellular organisms, and the conditions at the beginning of the experiment were the following. These cells were growing fine at 60 Fahrenheit or 15.5 degrees Celsius. They were really unhappy around 73 Fahrenheit or 23 degrees Celsius. And they were dead above 60 Celsius or 140 Fahrenheit. I looked up the weather in West Lafayette. It's about 90 Fahrenheit, so they would be really, really unhappy in there. But what William Dallinger did was the following. He slowly increased the temperature in the incubator and was always monitoring them, making sure that he doesn't kill the organisms completely. So over 7 years, he achieved the following. He made the organisms to grow at 70 degrees Celsius or 158 Fahrenheit. That's really hot. If you touch water that's that hot, you would get a burn. And some of these organisms who would be really unhappy in today's weather in West Lafayette could suddenly grow, well, not suddenly, but over 7 years of evolution could grow in scalding hot water. And their phenotype changed so much that when he put them back to their initial temperature, they could not grow anymore. So he made this very big phenotypic change from 15.5 degrees Celsius to 70. Another example, which is very famous, is Lenski's long-term evolution experiment. And this experiment is very big. He wanted to study several things, such as the dynamics of evolution, the rate of change, how repeatable is evolution, and what's the relationship between the changes in the phenotype and genotype. And for this, he did the following experiment. He started 12 cultures, 10 milliliters each, and every day he passaged them. He took 100 microliters and passaged them into 10 mils of fresh media. And he did that over and over and over again for many years. He's still doing it now. And it was started in 1988. And this is an old graph. By now, there are 75,000 generations. And here are some important numbers on the experiments. This is also an old graph. But some important numbers are this. They had 6.6 generations per day. We'll look in a second where this number comes from. They have more than 50 publications, and the cost was more than $4 million, so it's quite big. And yeah, they did a lot of cultures, as you can see in this photo with the plates. And something very interesting that they observed in this experiment was the evolution of a clone that could metabolize citrate under aerobic conditions. And they observed this because around generation 33,127, one of the 12 cultures was more turbid. It was denser. And they looked what happened there, and they found that there was a clone who had a mutation in the SSP gene. It had a promoter that appeared in front of it, which was activated under high oxygen concentration. So suddenly this SSP gene was expressed under high oxygen. That's why they could metabolize the citrate under aerobic conditions. SSP expresses a gene that transports citrate inside the cell and succinate outside of the cell. So it's an anti-porter. And because they froze many samples during this experiment, they freeze samples every week. They could look in the freezer and see if they could find this mutation in the earlier populations. And they could. Actually, 1600 generations before that, they could still find them. So the obvious question here is, why was that mutation present for 1600 generations, but they could only see it with the naked eye so late? Another experiment looked on this and made a slightly different environment where they made a specific selection for the metabolism of citrate under aerobic conditions. And actually they saw that this mutation appears in only 12 generations, and additional potentiation mutations occur within 100 generations, so much quicker, right? So what I want you to remember from this story is that selection for citrate usage was weak. So selection strength is something very important that we'll come back to a bit later, right? So take home message, selection is important. So let's have a look at more basic questions. What are the general limits of experimental evolution? For E. coli, for example, the most famous organism, microbe, the doubling time is 20 minutes. That means if you take one bacterium, in 20 minutes you have 2, in 40 you have 4, in 1 hour you have 8. And if you continue this graph, in 10 hours you have over 1 billion bacteria, and so on. So they grow quite quickly, right? That's 3 generations per hour and 72 generations per day. Why did we see that number 6.6 in Lenski's experiment? The reason is that exponential growth cannot continue indefinitely, right? You can't just have bacteria growing infinitely. It only happens as long as the chemical composition of the medium that they grow in is roughly stable. So they're in exponential phase as long as they have enough nutrients, as long as there are not too many byproducts that inhibit growth. Only then can they grow exponentially and really multiply like crazy. Right, and the 6.6 number comes from the dilution factor. They were making a 1 to 100 dilution, and 2 to the power of 6.6 is 100. So with only 6.6 doublings, they could replenish whatever was discarded the previous day. Right, so they were in exponential phase for a very, very short while. They can do 72 generations per day, but they were only doing 6.6. That's less than 10%. Right, so how can we increase this potential and keep them in exponential phase for longer than that? Right, so I mentioned the problem was that the chemical composition was changing. The solution to that is a device called a chemostat. What it does, it keeps the chemical composition of the medium stable. That's why it's called chemostat. And it was first published by this French scientist, Jacques Monod. It looks like that. You have a reservoir of medium that slowly drips into the reservoir of bacteria growing, and the excess is also pumped away. So you have a constant dilution going on that constantly feeds the bacterium with fresh medium, takes the waste away. But how can you control the dilution rate? If you make it too fast, if you let the tap flow through, it will just wash away the bacteria, right? If you make it too slow, then you won't use the entire potential of bacteria to replicate fast. For that, you can shine a light through the culture and measure how many bacteria you have there, right? That's called the turbidity. And a turbidostat is basically just a chemostat which has a feedback control of the density of the medium, right? Chemostat, then turbidostat. And the algorithm is pretty simple. You look how dense the culture is, and you slow down or increase the speed of dilutions as necessary, right? Another thing I mentioned was that selection is important as well in these evolution experiments. So we can control the dilution rate with the laser, with the light source, but how can we control the selection strength, right? So we want to make something like this. When they're happy, we kick them with a small hammer. When they get stronger, we kick them with a heavier hammer, and so on. So we want to increase the selection strength as they adapt. That's exactly what William Dallinger did when he increased the temperature of the incubator over 7 years. He looked every now and then, and if they were happy, he would increase the temperature by just a little bit. So how can we do that? We can do that either spatially. So this is an example of a plate where on the sides, here, on the left and right sides, you have just an agar medium where bacteria can grow happily. If you approach towards the center, the next lane has a dose of antibiotic that bacteria don't like. If you approach closer, they have 10 times the dose. Closer, 100 times the dose. And the central region here has 1,000 times the concentration of the first lane, right? So bacteria have an incentive to move to the center. They first adapt to 1 dose, then to 10, then to 100, then to 1,000, and over time they move towards the center. That's what you see here. There is a very famous experiment on YouTube. You can find that very easily if you already don't know it. Another way to adjust the selection strength is in time. So you don't have anything changing spatially. Everything is inside the file where it's mixed. But you have an algorithm that looks at the growth rate of the culture. And if the culture is growing happily, you can increase the drug a little bit. If it's not growing, you can decrease it a little bit or keep it constant. Right? So this is called the morbidostat. Because morbid is the prefix that means death. So you control how hard you want them to die. And the first morbidostat was published by Toprak about 12 years ago or so. And it looked like this. It had many vials, well, 15 vials, and 2 or 3 pumps for each vial. So there was a whole array of pumps, a lot of tubing, and the vials were inside of an incubator. It was the first modern experiment of this kind. Since then, there were many designs of the morbidostat that looked kind of like this. And then we decided to build our own morbidostat, and which we called Replifactory. So there were a few things that we didn't like in the existing versions of the morbidostat. We wanted it to be easy to autoclave. If you imagine having all of those pumps, having to disassemble them and reassemble them back, it's impossible. Autoclaving is not a feasible sterilization procedure. That's why people use different procedure for cleaning them. They were flushing them with ethanol, bleach, and sterile water, but that may not help against biofilms. If you have a thick biofilm, you have to actually scrub it to sterilize it, or to make sure that the ethanol reaches deep enough. So there are publications actually that complain about how easy it is to contaminate the Morbidostat and how hard it is to clean. So that was the first major point that we wanted to fix. To do that, we decided to multiplex the pumps, because you don't need 2 pumps for every vial. You can use you can share the pumps, as long as there's a way to control where the liquid flows. And the third point is that we used DFMA principles, so design for manufacturing and assembly. This is a big device where the components have a complex interplay, and there were many iterations of optimization to improve it. So something that we didn't really change with the years was the schematic. So it still looks like that. It's 3 pumps, 7 vials, and 7 valves that control the flow of media. I'm sure you understand this graph because you've used the devices. A small change that can be done to this schematic is with the pump number 1. You can make it pump from the first vial into the remaining ones. So you can feed a lagoon where phages and bacteria grow together. So you can have phage evolution experiments. That's something that I'm working on now. I'm trying to get the system to run well. But for the Morbidostat, we had very, very many iterations. It started off like that, and that's a rough timeline of what they went through. So in 2019, it looked roughly like this. The 3 pumps were here inside the box under the valves, and the valves, as you can see, had small white inserts, which you'll see in a few slides. Now, the problem with that was that to disconnect the tubing from the pumps, you had to take off the top lid. And that was kind of a pain. It didn't really make sense to have the pumps inside. So immediately afterwards, we hacked the devices to look like that. So the pumps were outside and the tubing was easily detachable. This worked better. So the next iteration, we changed the printed circuit board to be a bit smaller, from this green version to this black version, and made 3D printed parts that held the pumps and valves in the position that was comfortable. And that's something that Mike has played with, I think.
Yep, correct.
A bit later, we figured that it doesn't really make sense to have 3 vials here and 4 vials here in 2 rows. It's kind of annoying to have them interlaced with each other. So we made the obvious design choice to put them in a line. And the device looked like that. Yeah, and now you know how the device looks now. So how does the design process happen? What's the process of designing many iterations? So this is a short video of how a valve works. This is one of the earlier valves. It was designed in SolidWorks, which is a 3D modeling software. We used smaller motors, which were weaker. And the mechanism is like this. There is a cam that twists, that pushes the small door and pinches the 3 tubes. And you can control all of the dimensions in this model. So there are, these are in millimeters. And the drawing process is actually pretty simple. You draw squares and circles and triangles and put dimensions to them, cut them in different axes, and you build this kind of things. right? So once you have a 3D model that works well on the computer, you have to print it. And to print it, there's a two-step process. First, you have to give the 3D printer the instructions how to how exactly to move the print head to put the plastic in a way that makes sense. And that's what I meant with the design for manufacturing and assembly. There are parts that are easier to 3D print than others. For example, here I made the choice of designing this pincher so that the sharp edge is one line at the top. Yeah, as you can see, the nozzle moves in one axis, then goes to the next layer and continues depositing plastic from bottom to top. And if in the first version of the valve, this door was oriented vertically, so the edge was not very sharp because there were many layers along the edge. And once the slicer does its job, then you can give these instructions to the 3D printer that moves like that and pushes a reel of filament through a hot nozzle and deposits it to form any shape you want, really. Right. Next, electronics, also a major component of the device. This is the electronic schematic of one of the earlier versions that's on the left. That was the board in black that I pointed at before. This was a two-layer board. So you can see layer 1 at the top, layer 2 at the bottom. So there were copper layers on two sides of a non-conductive sheet. And the electricity flows through the wires between the microcontroller and other components that are soldered onto the board. One problem of the device that you can see here is, so at the top here, there are 7 connectors these are for the stirrers. So they were all connected in parallel. So whatever speed you gave one stir was the same for all of them. And that was really annoying because as you probably noticed, you had to calibrate the stirrers. Some of them like to spin faster than the others because there's more resistance in the system. So we decided to make a new schematic to fix that. And we fixed a bunch of other things in the process and realized that 2 layers were not enough for the complexity of the board. And yeah, the last version looks like this. It's a 4-layer board. So in colors, you have the different layers. There are red, green, yellow, and pink. And you can see that there are so many intersections between these small wires that it wouldn't have been possible to do it on 2 layers. And yeah, the board looks like that now. Guessing some of you might have opened the box to look what's inside. That's what's inside. Also on the website, you have a nice 3D model that is not mobile friendly, but from a computer, you can play around with the model and dig through and see all of the components, including this board. Yeah, so as a summary, we can use this device for science and education projects. For science, it's most notably used for antibiotics. So people study antibiotic resistance with Morbidostat. That's a very hot topic because it's medically relevant. My PhD project is looking at metal ions. So I want to look at how bacteria adapt to heavy metals. And I'm running experiments with nickel and cobalt. So I've adapted E. coli to nickel and cobalt, and I'm looking at what's the genetic response of this. And of course, phages. I'm working on making the machine run phage evolution experiments. I don't know exactly what I'm going to do, but that's still in development. And you can also do interesting experiments for measuring things that you can't otherwise. For example, yeast fitness in ethanol. Our group has a project with yeast in ethanol, and growth rate in plate readers works fine only for one growth curve. So yeast grows until saturation and then it stops, and you have a small window where you can measure the growth rate. But if you keep it continuously, you can have like a 1 week window where you can see how the growth rate changes, and it changes quite a lot. And that's something you can do in a continuous culture device such as ours. And for education, we used it at the School of Molecular and Theoretical Biology in 2019 in Spain. Maybe we'll use it this year in Tartu in Estonia. We had an interesting project where actually middle school students aged around 10 to 12 chose the parameters of an experiment that simulated patient treatment regimes. So students so young could actually understand the meaning of the experiment, and they could appreciate that if a patient takes the correct dose that was prescribed by the doctor, this is the least likely to lead to antibiotic resistance. But if the doctor prescribed 2 pills and the patient takes 1 pill, and then a couple of days later he realizes that, oh, it doesn't help, let's increase it to 2 pills, that's actually a very bad example. That's exactly the regime of the Morbidostat. You start off with the low dose, and as the bacteria adapt, you increase them. You increase it. And it was very interesting to see how really young kids could understand this principle so well, looking at the experiment that they programmed on this device. And of course at SSP, this year is the first time where 10 devices got used. I guess you can tell me more about that. I know it was rough at the beginning because getting something like this to work is not an easy task. But I've seen some nice graphs in there. I know something was beautiful. Yeah, so this is the end of my presentation.
We have time for Q&A. Can you hear me, Catalin? Very good. Not well.
I could barely hear you.
All right, I can stand up here and relay questions to people.
Yes. Or if you type them in, I can also see them.
You got questions? I can relay them, Catalin.
I've been waiting for 4 weeks. I know you have questions.
So I have a question. So we've studied antibiotic resistance and they're looking at genes for that. How does that compare to the resistance for your metals?
Okay, that's an interesting question. So antibiotics usually target one protein. That's very often, for example, the ribosome. And the antibiotics are really specific. Like it's this complex molecule that is designed to attack bacteria very specifically and not to damage human cells. So because of that, resistance usually occurs in like a few genes. It's maybe some protein that pumps antibiotics out of the cell. Maybe it's the target protein. So if you flip a few amino acids in the target region of the an antibiotic, it will just not bind and you have resistance. With metals, it's and that's why I find metals interesting, is that they damage cells in a more complex way. They damage DNA directly, they damage the membrane, they damage proteins. There are enzymes which depend on metals, and they can displace these right metals and make enzymes not function. So the selection response is more complex. It's similar to, for example, heat treatment, right? If it's too hot, you can't just mutate a couple amino acids in one protein and fix the problem. you have to really make sure that the entire organism survives and that all of the proteins do not boil. That's why I think it's interesting. And yeah, so and the genetic response, well, my prediction is that in antibiotics it will be yeah, very specific to a few proteins, and in metal ions it will be very general.
Thank you. Oh, Michael.
So, I'm not sure if you can hear this or if you'll
I can hear it very well.
Oh, okay. So, my question was, obviously making the chemostats and like Morbidostat has been a very like a long process. You showed us the 2020 version, the 2021 version. So what is like at this point the timeline going forward in terms of like finalizing the chemostat, in terms of dissemination of it? Like what is the next few years or however long that takes?
I've been meaning to publish a paper about the Morbidostat for a while. Yeah, we really need to sit down with Fyodor, my PI, and just publish this paper at some point. Regarding development, yeah, right now I'm working on this setup with phages. I think yeah, probably a big bottleneck now is software. There are some things that can be improved very quickly, which will make it a lot easier to use, and those are mainly in software. So software is pretty slow right now because the main focus was not to get it working fast, was just to get it working. And these are long-term experiments. It doesn't really matter if you wait 2 seconds or 10 seconds for like a small operation to run. So it was not high on the priority list, but now I feel that it's kind of getting annoying that it's too slow. Like, you know, the phone is working so fast, and here you press a button and you have to wait 5 seconds. It shouldn't be like that. In hardware, something that I regret not having done before SSP, there is a simple way to add flood detection. So if something overflows and the bottom of the device is covered in liquid, you can stop the experiment immediately. That's actually very easy to do. There are GPIO connectors here, general purpose input output, like these bunch of pins. You can just connect 2 wires in them and measure the resistance across them. And if there's a liquid such as LB, the resistance will decrease. You'll have a connection, and that will indicate that, oh, you know, something is running electricity through these wires. That's probably liquid. Stop the experiment right now. Yeah, that's something that I will definitely do probably with the next device that I build.
To do that without us telling you that was happening.
Yes, yes. Well, part of the reason why I made this pin was for that purpose. I'm getting this kind of floods too. It happens every now and then. There's no way around it. And that's normal for such technology in general. Like the first iterations, you know, they have some hiccups. You have to really make sure that everything is perfectly fine. You know, if a small thing is not right, it fails.
Okay.
But yeah, probably in a few years it will get to the point that it will feel like an iPhone. You know, you order it and it just works. You don't have to think about it.
Good. Another question?
Yeah. So I know you had mentioned that like a general problem with Morbidostats that's been discussed is like contamination, and I know that was that a lot of us experienced. I was wondering, is there any way, like anything that you're looking into, or any way in general with Morbidostat to decrease that risk?
Yeah, I think well, contamination is usually from the environment. I think, yeah, having to put the bottle caps onto the medium with the bottle with medium is like the riskiest step. One way that I was controlling this at the beginning, when I was getting way more contamination than now, was to feed growth media into the bottle with a needle. So I have another bottle that I autoclave, and I just place it somewhere high and start pumping the liquid through a silicon tube that has a needle at the end, and I inject this needle. I put this needle into the cap of the main bottle. I think this is confusing. So basically, you don't have to open the lid and put it onto a bottle. You put a needle into the lid, and a slow stream of media flows in. So that would mean that when you autoclave your medium, you autoclave it with this special system outside of it. Yeah, I think that's the only thing that can prevent external contamination, because it's already so much harder to contaminate than the original Morbidostat because it's autoclaved as a closed system. Right, so you can go around, you can sneeze on it, and it shouldn't get contaminated. But there is still this last step where you have to open the lid and get it to start.
Yeah, you mentioned that in this model, you had one bottle connecting to all of the vials instead of previous ones where you had multiple bottles or a bottle for each vial. Do you think that possibly could have increased the risk of contamination?
Could you hear that?
So what that increases is the risk of cross-contamination. So this makes it possible for culture to flow through the tubing from here to here. And if that happens, the seventh vial is contaminated. So one of the vials is kept as a control, so that if something reaches this, well, sorry, you couldn't see. So it increases the risk of cross-contamination from here to here. So if there's medium reaching this common area, it can go to another vial. And you control for that by having one vial where you don't inoculate anything. And you know that if something starts growing in there, then you have a contamination problem. But it does not decrease it does not increase the risk of contamination from the bottle. Because in the original Morbidostat, the bottle looked, wait. Here. So the medium bottles, you can see, have a lot of tubing inside, because every single vial has its own tubing that comes here. I think sharing them would be a bit better. But having to put that cap on the bottle is even more risky, because you don't just have one tube. But I think that's absolutely normal that you had contaminations. You guys have been working with the device for like 2 weeks, and I was getting contaminations for the first year. So it really takes some practice to get all of the small steps, but that's normal, and that's already such a success of having a summer school run this kind of experiments. So Erdal Toprak has a lab with these devices, and he said that he actually has a technician working full-time just to run this. So he's doing science experiments on them, but there's one technician whose only job is to run this machine. So having 10 machines is like quite a high goal that we set. And I think a little bit of contamination is really acceptable for this.
So you mentioned how you're also studying the effects of metal ions and phages on bacteria. So what other avenues of research do you think are made possible or made better by the use of the Morbidostat?
I keep thinking about growing mammalian cells. So you can have something like some leukemia cells that grow in a liquid culture, and you can have evolution on like human cells. I've never done it, but I think it would be interesting. It's so this would be not a lot easier than doing it by hand, because human cells just grow so slow. You can as well just come and kind of pipette them out every day. But yeah, I keep thinking about setting up a device where like just human cells grow and then forget it for 2 months and then come back to it and sequence it and see what evolved in there. And that's something that you can do only if you have a very sterile environment. So I would not do that if the sterilization method was like flushing with ethanol. You really have to autoclave that medium. Because they grow so slow, anything that remains there would break the experiment. And in general, I'm fascinated by the idea of playing with liquids, of pumping them back and forth and making biological systems interact with that. Yeah, I don't know. I'm guessing you might have ideas that can be built with that. I would be happy to hear about it. On replifactory.com, there's a forms tab where you can like write in suggestions and ideas.
So I know that with studying like diseases that are applicable to the clinical scene, for example, one of the big issues is studying things in an enclosed environment versus studying them in like out in the wild, because they have different factors that affect them. So is it at all possible to like modify the Morbidostat, for example, to better mimic the environment in a way that we haven't before?
So what you can do, I suppose, is use, yes, so something closer to the environment would be, for example, to take not just like one species of bacteria, but the whole population. Like sample some population of bacteria from the pond or from soil and grow it in a medium that resembles natural conditions. So LB in principle is pretty close to that, although it is a kind of controlled lab environment. But I guess the whole idea of the Morbidostat is not to mimic the environment, is on the contrary to control it. Yeah, I guess the beautiful thing about the stuff happening in the environment is that you have fluctuations of all kinds of things, like the temperature, the amount of light. With seasons, you get rivers washing away different kind of salts, and you get different populations increasing depending on the season. Like in winter, those guys grow; in summer, the other guys grow. I don't think it's worth trying to replicate kind of the nature outside in a test tube. I think the interesting part about the Morbidostat is that you can take something from the nature outside and look at something very specific, because you have the ability to control something about it.
Something we talked about was like that we had to consider some of the mutations we got were for fitness growing in the chemostat rather than like actually resistance to the antibiotic. So I was wondering if you have anything you can impart on us about that possibility when using a Morbidostat.
Just anything you can impart on them, any knowledge about kind of how things will start to grow in this environment specifically.
Well, you've used Vibrio nitrogens, right? That is a lab organism. So it has been growing in the lab for many years by other scientists. So it has already undergone some rounds of selection for growth in LB in the lab. I think the biggest, the most important source of mutations that you should see with an increasing dose of antibiotic is really just because of the antibiotic. If you took something from outside and started growing it in LB, it would need like the first optimization process that it would have to undergo was first, let me learn to grow in LB at this temperature in this shaky flask environment. And only after that, you can think about adding a bit of antibiotic and looking at how it will change to surviving with the antibiotic. But Vibrio nitrogens is like a well, more or less common lab organism. So it already has most of the mutations required for living in the lab. I think, yeah, for a lab organism, I think this shouldn't be the biggest concern.
What are your thoughts on pumping in another species instead of antibiotics?
Pumping in another species for what?
Like instead of using antibiotics as a challenge, and instead you would like pump in another like strain.
Yeah, so that's what I want to do with phages. So with the I can explain a bit the idea. So what I want to do is host shifting in phages. So you have a phage that can infect species A, right? And you can give it species A every 2 hours, and you can see how it's killing it and growing well. But then it cannot infect species B, and I want to make it infect species B. So what I can do is take like run this continuous culture with species A. So I have a very large population of phage that I can play around with. And what I could do is inject something in the culture that kills all of species A, and then I add species B. So now we have a very big population of phage that has a selection towards infecting species B. Right? There's a lot of phage that would be happy to reproduce in a new ecological niche, and we can do this automatically. So we add this species B. If it works, great. If it doesn't, it will just start growing and the phage will not do anything. But if it doesn't work, we can automatically flush away all of the liquid, the mixture, and start again feeding with species A so that we replenish the phage population. And I think this is how it works in nature. It's not enough for selection to be there. It's also important that you have a large population that is under the selection. For example, if you have bacteria, there are bacteria that grow in crazy environment, like in salt mines and metal mines. So that there are bacteria that can metabolize metals because they have been growing near metal mines. And they got this ability not just because they were close to the metal mines, but because it was a lot of them. So out of the millions of bacteria that were near the mine, some of them had the lucky mutation that let it use this new niche, and now we know about it. So with phages is the same. I think if you do it once, like if you take one phage and put it in species B, probably it's not gonna work. But if you take 10 billion of them and do it multiple times a day, it increases the probability that, oh, maybe at least in one of them, there will be this mutation that will make very novel functions.
Any more questions? All right, so for these 16 and 17-year-old future scientists and doctors and everything else, do you have any final words of wisdom as they start on this journey?
That's probably something that I wish I would have heard when I was their age.
I didn't prompt him with this question earlier, so he's coming up with something on the fly.
No, yeah, so I think, yeah, as I've showed in the presentation, there were a lot of things to work on with the Morbidostat. And when I began, I had very little experience with most of them. I'm very happy that I got onto this path, because I've learned a lot of things very quickly, because I had this project, I had this dream of building this kind of device, and no matter what, I wanted to solve it. I think that's really important, and if you can find something like that, it will be a lot easier than sitting in a boring lecture and listening to some theory being told, like this one.
All right.
Yeah, it was really interesting to improve my coding, my 3D printing and 3D modeling skills. I started off with nothing. When I think about like 3 years ago, I was doing really stupid designs, and then I was learning how to do it better. But you know, looking back and cringing is the sure sign of growth.
All right, so with that, let's give him a round of applause. Thanks for taking the time to speak with us, Catalin, and I'll talk to you, before next year.
Yeah. Thank you for the opportunity, and thanks a lot for using the device. It's really a big success for me to see this big room of people who use these toys. Enjoy the rest of your school.
Yeah, have a good day. Just